Welcome back, ladies and gentlemen. This is screencast number 2 of 4 for genetic engineering. This topic is going to be on manipulating DNA, and the reading that you should have done prior to this screencast is section 15.2 in your textbook. So if you haven't read that yet, go ahead and pause this, grab your textbook, take a look over the section, and then revisit this once you've read through it. This document is in Google Drive. It's also linked into Schoology, so if you ever need to access the actual PowerPoint to study from, you can access that there, and the screencast is embedded right into that lecture. So let's get started for today. Today's topic is on manipulating DNA, and I first need to clarify that usually when we hear the word manipulating, we mean talking someone into something, like manipulating people to get your way, or manipulating being a bad thing. However, in its actual meaning, manipulating means to change or to sway or to actually have a hand in actively changing. So when we talk about manipulating DNA, we simply mean to change it. And there are 5 different ways that we can change DNA. The first way is that we can extract it. So we can take it from the cell that it belongs in normally, or the cell that it's replicated in, and we can extract it to make a solution of just pure DNA from that cell. Second way is we can cut it. So we actually use these things called restriction enzymes to cut it into little pieces, or to cut just a section of it that we want to work with. We can actually manipulate DNA also by identifying gene sequences. So we can look through an entire strand of DNA and try to locate a specific region on that DNA. Another way we can manipulate DNA is by making copies of it, and the last way is by recombining it. So taking a strand of DNA and splicing it with another strand so that we get a brand new strand of DNA. Okay? So let's start with what we know, and that is DNA extraction. We did this lab about 3 months ago when we took a strawberry, we mushed it, we put extraction buffer in it, we filtered it, and then we used cold isopropyl to actually make the DNA rise to the surface. And that process was meant to smash open the cell walls, open up the membrane, make all of the cells kind of mush, and then filter out all of the organelles so that the DNA would isolate up to the top layer of the interface of isopropyl alcohol. So that entire purpose was just to make the DNA separate from the surrounding cells that it's normally encapsulated in. So that's one way we can extract it. Keep in mind that if we did this with human DNA, we would need to take some sort of human sample. So we might take a piece of hair or a swab of our cheek cells or some sort of skin sample, and we have to extract DNA from that. Okay? Second way we can manipulate DNA is by cutting it. And like I said, we use these things called restriction enzymes. And I always think that restriction means that you stop it or you cut it. So restriction means like cutting enzymes. And enzymes, if you remember, are protein molecules that are many different amino acids put together to make a protein, and enzymes work to either build or break chemical bonds. In this case, if they are restriction enzymes, they're going to be breaking the bonds between DNA so that they can cut DNA. So I like to think of them as our chemical scissors. Restriction enzymes will create fragments or pieces of DNA that we can then work with. Commonly, we'll use restriction enzymes with plasmids, and plasmids are circular DNA that are found in bacteria that we can actually cut out of bacteria and insert into different organisms, and we can do some pretty cool stuff with that. There'll actually be more information with plasmids and bacterial transformation in the next screencast, so stay tuned with that. Down here we have a picture of restriction enzymes. So this strand would be our double-stranded DNA helicase here. And the restriction enzymes will cut between genes. So this section of DNA right here is going to be gene A. This section of DNA right here is going to be gene B. So remember, a gene is just a section of DNA that codes for a protein. The restriction enzymes are enzymes that are going to cut between those two genes. Okay, so this is kind of a cartoony example. Let's look over here at what the actual DNA strand would look like. So here we have our double-stranded DNA strand, and we're assuming that between each of these nitrogenous bases coming out in the middle of our ladder of DNA, there are going to be hydrogen bonds. If you remember, there are 3 hydrogen bonds that connect a G to a C, and there are 2 hydrogen bonds that connect an A to a T. So here in this step, we're seeing that there's this giant restriction enzyme. We actually call it a restriction endonuclease, but I usually say restriction enzyme. This enzyme is actually called EcoRI, or EcoRI. Kind of a weird name, but it's named after the bacteria that it comes from. So this restriction enzyme is going to jump on this DNA strand. If you notice, it's going to land on a section that is AATT. And if you remember anything about our DNA unit, A and T bond together with 2 hydrogen bonds, whereas G and C bond together with 3. So if you're smart, you're thinking, well, hey, if there's only 2 hydrogen bonds between this entire row of bases, this makes a weak point in the DNA. And that's exactly what it does. This enzyme will find that weak point in the DNA where there are all of the double hydrogen bonds between A and T, and it will actually snip right through those hydrogen bonds and break them apart. What ends up is not actually a clean cut, so they don't actually just slice down through the strand. You actually make what's called a sticky end, where they go down, over, and down again. So it makes these kind of tail-looking things, where it's a strand of sugar phosphate backbone with just these nitrogenous bases sticking up on either end. And you can see here's our familiar 3' and 5' end, which means that there's a phosphate group here, and this one ends with a sugar down here. So you might say, well, why do we want to cut DNA? What are we going to do with that? Well, we use restriction enzymes to cut DNA in different ways. So we can use different enzymes to cut at different restriction sites. So the enzymes will seek out an area and only cut in that specific area. So here we have a strand of DNA, okay? And in this diagram, it is the exact same strand of DNA. The only difference is that in this picture, we're using restriction enzyme number 1. And restriction enzyme number 1 is going to find a restriction site right here, and it's going to cut. It's going to find another one here and another one here. And if we cut 3 times, we're going to end up with 4 different fragments of DNA. Okay? So let's back up a little bit. If we would have that original same strand of DNA, but this time we would use a different restriction enzyme or a different pair of chemical scissors, it would actually bond to more sites. So 1, 2, 3, 4, 5, 6 cuts, making 7 fragments of DNA. So the cool thing is that we've learned enough about these restriction enzymes that we now know what restriction enzyme to use to get different sized pieces of DNA. And that's very, very useful for our genetic engineering. Now, why do we want to cut these little pieces of DNA up in the first place? And that is because we can separate DNA sizes by a process called gel electrophoresis. It is a really cool lab, and if you stick around for AP Bio, we actually do this lab, so it's pretty fun. Gel electrophoresis is a process that you guys will need to be familiar with. So if my explanation doesn't do it justice, I would suggest you come down here and click on this link. This is a link that takes you to the electrophoresis virtual lab where you actually go through it yourself and you'll see the results that you get. So the basis of electrophoresis is that we're trying to separate DNA fragments by size. Now the easiest thing would be just to look at them and figure out which one is bigger and which one is smaller, but DNA is so small of a molecule that we can't see it, obviously. So what we do first is we cut that DNA sample with restriction enzymes. So we take an enzyme that we know of. So maybe, for example, the one that we looked at before, the EcoRI enzyme, and we cut the DNA up into a bunch of little pieces. But what that means is we have a bunch of different sized pieces and we want to separate them to see what we have. So what we do is we actually put the DNA into Jello. And it kind of looks like Jello. It's not actually Jello. But we say that we run it through a gel. So we actually put the DNA into these little tiny holes in the gel. And you can see in this picture, there are little tiny indentations. We take a pipette and we suck up some DNA and we squirt it into these little wells. And what happens is we put a current, an actual electrical current through the gels. And what happens is that the part up here is going to be a positive current. So we attach a positive electrode up here. We attach a negative electrode up here and a positive electrode down here. And what happens is that the DNA that we put up here in these little tiny wells is actually slightly negative in charge. So you guys should know by now that negative and a positive charge always attract towards each other. So what that does is that in each one of these little lines going this way, that is actually DNA that is being pulled towards the positive cathode down here, okay? So what essentially we're doing is we're pulling DNA fragments through a really thick jello, and we're trying to see which ones can get to the bottom of the gel, this side over here, first. Now, if you can imagine having a race through a pool of jello, it would be really hard to move through it, and it'd be an advantage for you if you were a small person. So if you had a little tiny kid racing, a little tiny skinny kid that was racing through a pool of jello, he'd have an advantage because he could kind of squish through it faster. Whereas if you had a person that was huge, like a big linebacker on a football team, big guy, it would be harder for him to move because he has more resistance. So the idea is that as we pull all of these DNA fragments that we put in here through the gel, the small ones are going to zip down to the bottom really quick. But the big ones are going to be stuck in the gel, and it's going to take them longer to pull down. So what happens is the smaller fragments move faster, they travel further than the larger fragments that move slower. So what this means is that once we detach the electrical supply, that the fragments of different sizes appear as bands. They are frozen where they are in the gel. We can dye them a color so that we can see them, and they make these band patterns in the gel. At this point, if you haven't gone and done this virtual lab, it helps clarify a lot of this process when you actually see it for yourself. So what do we do with this? So if we get this result of all of these bands, how can we use it? And the reality of how we use this is we use it in DNA fingerprinting, which means we've talked about how every individual has a unique DNA combination of genes, which means my genes are different than your genes, and your genes are different than your mom's genes, that are different than her mom's genes, that are different than her sister's genes. Everybody's different. So if we can separate DNA, we should be able to see that everybody's DNA is different. If we run gels through, or if we run DNA through electrophoresis, it means that the results of one person should be different than the results of another. So DNA fingerprinting is when we identify people at the molecular level, meaning we look at their DNA, we run it through electrophoresis, and we see who they are. It's kind of crazy stuff. So how do we do this? How do we use DNA to fingerprint? Or how can we use DNA fingerprinting? We can do this as evidence in criminal cases. So we can actually collect DNA from a crime scene. We can use gel electrophoresis to run it through this gel, figure out who they are, and then match it with criminals that we've arrested by taking DNA samples from them and seeing if their DNA matches. We can also use it for paternity tests. So if we don't know who the daddy is, we can take DNA samples from the child, we can take DNA samples from the father, and then we can see whether or not they're similar or different and whether or not the father is the true father. We can also use it to study biodiversity. So right now, if you'd have the aspiration to become a biologist and work out in the Ecuadorian rainforest collecting different species of bugs, one of the things you would do is whenever you collect a new species, is you would study the DNA of that species to try to decide what makes it work the way it does. And you would use DNA fingerprinting to kind of figure out what is the DNA that is in that new species that you discovered. Okay, so I told you before, I asked you before, what is going to be the result of that gel electrophoresis that we were talking about? And this is actually a picture that they took of one of the gels. So let me explain what's happening here. In this little up and down column, this is DNA from the mother. So we took the mother's DNA, we extracted it, we cut it using restriction enzymes, and we ran it through gel electrophoresis. Each one of these bands represents a strand or a section of DNA found in this mother. Okay? Now, if we jump over here, this column is the column for the father. So each band represents DNA that is found in that father. Notice, if you compare against mother and father, the father's missing a band that the mother has. And that's because mothers and fathers don't share genetic information. They had different mothers and fathers than each other, so they're going to have completely different information. Now the difference is when we look at the children. So if this is the mother, this is the father, these are the rows for child 1 and child 2. What we're going to see is that we know based on genetics that every child either gets its trait from its mother or from its father. Which means that this child here, if you look at this band, this first band, each section of their DNA either comes from mother or from father. So this child got this band, this section of DNA from his mother because it matches straight across. If you go over this direction, if you're looking at this band, the father does not have that band, which means that whatever genes lie on this part of the DNA, child number 1 got that from mother. And so did child number 2, because here's that band here. Okay, now let's go to the next band. This band was actually present to mother and to father. It looks like child 1 got it, child 2 did not. So this might have been some sort of mutation that child 2 actually didn't get this genetic disease, or I'm sorry, this genetic strand of DNA. If we go down to the next band, here we have a band in child number 1 that is not here in the mother's DNA. So that means we need to look to the father, and there it is. So the father right here had that band, so obviously child number 1 and child number 2 got that band from their father. The 4th band down looks like everybody has, okay? The 5th band, look at this. The mother has, but child 1 does not, but child 2 does. So what that means is that child 2 got a trait from his mother, and child 1 didn't get it, okay? Which is kind of crazy. So child 2 would resemble his father more for that particular trait. Okay, if we jump down here, this is cool too. Here we have the band that child 2 has that his father has, but child 1 does not. So that means that he got that piece of DNA from his father. Okay, so DNA fingerprinting depends on the probability of a match. So basically, what is the probability that an entire section of our DNA is going to match somebody else's? Many people have the same number of repeats in a certain region of DNA, but the probability that 2 people share that exact number of repeats is exceedingly small. So that's the reason why this whole process works. Now, I've embedded a little puzzle for you guys. This crime you can read down through, and you can actually solve it by going through and going to this website. This website at pbs.org takes you through analyzing the fingerprint of this mystery of the crime. So it's kind of a cheesy example, but it does do a nice job of showing you how this process of gel electrophoresis takes place. So I would suggest read the crime, go to the website, take some time, figure out how to do a DNA fingerprint. Last thing I wanted to end with today is just to give you some examples. We can use DNA fingerprinting to solve crimes. So take a look at this. We have a crime scene DNA column here. So this is the results of electrophoresis. Here we have the column. So this would be DNA that we collected from a crime scene. So maybe we found a fleck of blood, or maybe we found a piece of hair from the crime scene. And we're trying to figure out whether the DNA, this band, this band, this band, and this band, is going to match either one of these 4 suspects. So what we do is we have all 4 of these in custody. We take a blood sample, or we have them spit in a cup, or we take a piece of their hair, and not their hand, that would be weird. We take a piece of their hair, we extract the DNA, we run it through gel electrophoresis, and we figure out what bands they have in their DNA. So the way that you solve this is you look straight across from the crime scene and you see whether or not the bands perfectly match with one of these 4 suspects. Okay? Right away, I'm seeing that darling Miss Daisy probably is not our suspect, because take a look. At the crime scene, whoever did it had this band of DNA, and that band is missing in Miss Daisy. So I'd say right away she's probably not a suspect. Okay? But Bugsy, Dino, and Tootsie all had it. All right? Let's move on to the second band. The second band was actually found by all 4 of them, so probably not going to be them. The 4th band down. Again, this 4th band is found in all of these victims, so probably not telling us much about that. And the 4th band down is going to be found only in Dino and Bugsy. So that tells me that if it's not found in Tootsie, I can eliminate her as a suspect. So Daisy and Tootsie are out. It was either Dino or Bugsy. I don't know who came up with these names. So let's look at whether Bugsy's DNA is closer or Dino's DNA is closer. And it looks like right here, Bugsy has a band that was not found in the DNA of the crime scene. That is a huge red flag, and it looks like Bugsy was not our suspect. So in this particular answer, Dino was our suspect. All right, here's another example of a whodunit. This is the same gel electrophoresis, but I want you to see that the banding patterns are just shown by the blue lines instead of the ones on the previous example. Over here, we have the victim's DNA. So it's always important to gather the victim's DNA so that we can determine whether the samples that we get from a crime scene are from a suspect that we need to investigate, or whether or not it's from the victim itself. So if there's blood at a crime scene, we can't automatically assume that it's the suspect. We have to rule out the fact that it's the victim, okay? So in this particular example, we have the victim here, we have suspect 1 and suspect 2. And then here we have one sample from crime scene 1 and one sample from crime scene 2. So what we're going to do is we're going to see who was at crime scene number 1. So crime scene number 1, let's look at this. If we go down, our first band is right here. Found in both of our suspects, doesn't tell us anything. Crime scene band number 2 was found in this suspect, but not in this. So ladies and gentlemen, it's looking to me like this suspect number 2 was at crime scene number 1, and that's a huge indicator. Let's double check the last one. Yep, here we have a band in this one and a band in this one. So suspect number 2 would have been at crime scene number 1. Now crime scene number 2, if we look across here, the only band that is found in crime scene number 2 is not found in the suspects, but it's found in the victims. So what this tells me is that crime scene number 2, the DNA sample that I got from it is actually a sample from the victim, not from the suspect. Okay? So the last way that we can use DNA fingerprinting is to determine paternity. So right here, we have an example of when a soldier unfortunately died in combat, and this unnamed soldier needed to be identified. So four parents came forward saying that they didn't, that they can't find their son, that he didn't show up from battle. So we're trying to figure out which parents match the soldier's DNA. So our options are parents A and B, C and D, G and F, or E and F. I'm sorry, G and H and E and F. So if we look across, we know that a person like this soldier is either going to get a band from its mother, or from its father. So here, the soldier has a band that is not found in either of the parents A and B. So that tells me right off the bat, parents A and B, probably not the parents of the soldier. Let's look at that first band, go over to C and D. Well, here, parent D does have that band, so it might be C and D. Let's go over one more. Here, that band is still found in parent E, and it's actually found in G and H. So right now, I think that it's down to these 3. If I go to the next band here, this second band is found in parent C, but not in parent D. If these 2 are parents, it means that that soldier could have gotten it from here, so it still might be parent C and D. That same band is found right here, so it still might be parents E and F. But that band is found here on that parent H. All right, so right now, I'm not sure who it is still. If we look at the third band, this really, really thick one, we can see that it's here in parents C and D, here in parents E and F, but it's absent from parents H and G. So now we have it narrowed down to parents C and D and parents E and F. Let's keep moving down. I'm seeing 3 bands. So let's look at this first one. If I go straight across, here that band is in parents C and D. So right now they match up. But look straight across, that is not found in parents E or F. So I think we just solved it, guys. If this band is found in parents C and D, it means that that soldier had to have gotten it from these parents, and therefore they are the true parents. But let's double check the rest of them. This middle band was found right here. The last band was found right here. Here, this band is found in this parent. This band comes to parent D. This band comes from parent D, and this last band comes from parent C. So it looks like parent C and D are our correct parents. Okay, nice job, guys. This is the end of the second screencast in this sequence of genetic engineering. Stay tuned next for topic 3, which is transformation and GMOs.